Thank you.
Nice. So a bit about me. I'm a fourth-year apprentice doing my applied bioscience at the University of Kent, and then I work at AstraZeneca as a technician scientist, and I work in the in vivo team in the early oncology bioscience department. And then I live in Luton, I work in Cambridge, and I enjoy playing golf and watching Luton football. So a bit about the in vivo team in the bioscience department. So the preclinical in vivo team sort of falls in this stage of drug discovery. So there's the target selection and target validation. And then once they've found some compounds that they think may work, we screen them internally in vivo. We potentially make financial investments to expand our project teams if there's potential. We submit publications, and then we choose one compound to take forwards and invest potential combinations. And then even though it's moved on from the preclinical stage and it's in the clinic, we still continue to do work preclinically to understand the compounds more. So a bit on what is in vivo work. So we implant tumor cells into mice and sometimes rats. We dose compounds at various concentrations, and these compounds can be dosed orally into the vein or into the abdomen, directly injections. And we're trying to understand the impact of the compound on tumor sizes and potentially any biomarkers that we think may be targeted by the compound. We assess any toxicity of the compound. Then we can take samples either during life, we can take blood samples through the vein, or then we can take terminal tissues and blood samples at the end. And I think it's really important to stress every time I talk about it that a drug cannot be tested in a human without prior safety and efficacy in another organism, and it's currently essential as part of the delivery of new medicines to humans. So there's various types of in vivo studies. This is a big boring table showing all the types and what they do, but I'll summarize all of them here. So a growth curve study, if you've got a compound in oncology, you want to test it on a tumor cell line. So you need to get the tumor cell line to grow in mice first. We use these studies to test the properties. So how quickly does it grow? Does it grow variably, or is it nice and consistent? We typically do 5 mice per growth condition. So a growth condition can be which sex of mouse, which strain of mouse, how many cells per mouse, do you grow it in PBS, MHGel, that sort of thing. And you're just trying to find the optimal growth conditions for what you want to do. So you don't want it to grow really slowly and do nothing for 80 days and then grow at the end. But you don't want to, likewise, you don't want it to grow really quickly. So this is what like a nice growth curve would look like at the end with the tumor volume and the days of study, and that's what a bad one would look like. So the difference here was the strain of mouse, so a C57 mouse versus a NSG mouse. And the difference between those two are C57s are fully immune competent and these are fully immune compromised. And so this was likely to be a human cell and not rejected by the immune system in NSG mice. Tolerated dose studies, so we test that the compound is tolerated. And this is required for all new compounds, or if you're trying to use an old compound at a higher concentration, or if you're going to try and dose it through a different route, or if you're going to try and combine it with something else. These are typically 2 to 3 mice per group, and there's no tumor, but we weigh the body weights of the mice throughout, as the body weight is our key quantitative way of measuring tolerability. And so this is what compound that's not tolerated would look like. So by day 16, it had lost 15% of its body weight lost 15% of its body weight. And then this is a compound that's tolerated quite well and it doesn't lose any weight. And yeah, so that was compound A tried to dose at 50 milligrams per kilogram, whereas this was 25, so half the dose. And then once you've got a compound that is tolerated and you've got a model that you want to dose with the compound, you'll do a pharmacokinetic / pharmacodynamic study. And so pharmacokinetics are the way of working out how the compound reacts in the body, and the dynamic side of it is how it reacts with the tumor. So we would dose mice that have got tumors implanted into them, and we'd dose them with the compounds. They can be dosed once in an acute study or chronically for a week or week or two. And then typically at the end, we'd collect bloods and we'd collect tumor samples, which we can assess. Blood samples can be used to find out the concentrations of the blood, find out the concentrations of the compound in the blood, and tumours can be analysed for a variety of methods. So it could be Western blots, it could be a mass spec image, and we can. So this is an example where we were measuring the levels of estrogen receptors in the tumour when dosed with various compounds, and so we can see compound G and compound H degrade the estrogen receptor really nicely. And then a full chronic efficacy study. So we're dosing the mice typically for 4 to 6 weeks. We'll have various group sizes. So if it's a really variable model, we might have 10 to 15 mice per group. If it's a really tight growing cell line, we might only need 6 or 7 mice per group. And typically a study would run with lots of different compounds, or one compound at different concentrations, or you could combine multiple compounds. And so this is the relative tumor volume. So it's the size of the tumor compared to its original size on day 0. And as we can see, the control it grows upwards, it increases in size, and some of these compounds down here actually make the tumor shrink, which is exactly what we want to see. So as with all animal studies, there are ethical considerations with everything that we do. So even though it is essential, we try and do everything that we can to minimize the amount we actually do. So where possible, using cells in flasks rather than cells in mice. All of our studies are powered, which means that we're getting statistical relevance from all of our data, but we're not using any more than we actually need to. So if we need 10 mice, we won't use 12, but we also won't use 8. Only studies that are scientifically valuable will ever be run. And at the core of everything we do with animal work, there's the 3 R's. So reduction, refinement, replacement. So that would be reducing the amount of animals or the amount of animal studies we do. Refining, so improving the welfare and the life of the animal while on study, and then replacing it with alternative methods where possible. Everything that we do in terms of a group, and me as an animal technician myself, we're held accountable by the Home Office and the local ethics board. And so at least once a year, we'll be visited by the Home Office just to see what we're actually doing day to day. And if anything goes against what we said that we were going to do, it's reported immediately. And then public perception of our use in animals is ever changing, but we're always trying. We've got a big openness policy, so we try and educate people, whether that's through schools, science fairs, that sort of thing, just to drive further understanding. Because I think a lot of the concerns about animal welfare are due to lack of understanding of what it actually is and what we do. And then a little bit of a case study towards the importance of in vivo work. So I work on a project called capivasertib. So this is capivasertib, the chemical compound. It's a small molecule. It inhibits AKT, which is the central node in the PI3 kinase AKT pathway. It acts as an ATP competitive inhibitor of AKT, and by inhibiting AKT, you prevent the phosphorylation of downstream substrates. It's dosed orally. It's dosed typically at 130 milligrams per kilogram twice a day using an intermittent dosing schedule. So we dose it for 4 days, then we don't dose it for 3 days, and so on. It's got some side effects, including hypoglycemia, so high blood sugar levels, diarrhea, rash. And then, amazingly, in 2023, it became an approved medicine and is now called TrueCap. So, preclinically, we understood that capivasertib, or back then AZD5363, we could see that it had efficacy, but it was quite difficult to get it tolerated. So when we dosed it in mice and rats, the animals often lost a lot of weight and they showed clinical signs. So we tried dosing it twice a day at a lower dose versus once a day at a high dose, and we tried dosing it every day versus some days on, some days off. And we found that a lower dose twice a day and then intermittently, so 4 days on and 3 days off, you could actually provide equal amounts of effect compared to a high dose once a day. And this improved the tolerability substantially. And so we always say that scheduling is crucial. It could be the difference between a drug that works really well versus a drug that causes harm. And so a graph here showing this and then expanded down into here. So we've got 300 milligrams once a day, in the, 300 milligrams 4 on 3 off versus 100 milligrams twice a day continuously showed improved efficacy compared to continuously while providing similar amount of compound over a week. And so that showed that actually taking breaks in between doses can improve the body weights. And then this is a graph here showing, again, continuously twice a day versus 4 days on, 3 days off, and the weight loss was substantially smaller. And so why was that important preclinically? Well, we did a very similar study in the clinic showing different dose ranges, some continuously dosed, some 2 days on, 5 days off, some 4 days on, 3 days off. And we actually found that doing the 4 on 3 off in the clinic had a similar effect to what we saw in the preclinic setting. And so 480 milligrams, 4 days on, 3 days off, twice a day was taken forwards. And amazingly, after that scheduling in phase 1, phase 2, and the preclinical setting, after a phase 3 trial, capivasertib was approved. And we found that in ER-positive breast cancer, for those that also have PIK3CA, AKT, PTEN mutations, capivasertib added to fulvestrant had a 50% reduction in death or disease progression versus fulvestrant and placebo, shown here. And if it wasn't for the preclinical work, capivasertib wouldn't be TrueCap as we know it today. Are there any questions?
Fantastic. Thank you, James. There's a few questions in the chat, so I will just try and let's go back to the first one. So the first one is, what have we got here? How do you calculate the tumour volume in Vivo?
It's not very fancy at all. We've just got calipers. So yeah, we get calipers and we just measure the length and measure the width of the tumour, and then we input it into some software that we've got that calculates a 3D volume. Yeah, not as fancy as you think it might be.
And we have another one from Claire. So what are your thoughts on the 4th R responsibility?
I'll be honest, that's not one I've heard of, but I can imagine, yeah, responsibility, being responsible for everything that you do, but also being responsible of the animals when they're in your care is, yeah, really important.
Can I just jump in on that one, James? Because I was thinking about this when you were talking about it. Because my project is kind of got a sustainability angle to it, and I thought it was really interesting to hear that you said you're spreading awareness, because I think a lot of the issues come from ethical issues come around people not understanding what we already do, right? What all the lengths we go to when it comes to whether it's animal testing or animal-derived reagents in my case. So I really like that you said you're spreading awareness from that point of view, but could you remind us what you said you were doing? Was there some sort of forum you were part of or a group?
Yeah, so we're trying to spread awareness both internally in AstraZeneca, because we're such a massive company. There's people that we sit next to every day that have no idea what we do, even though we work in the same building. So we're trying to take everyone that we work with directly, and then also those that we work indirectly with, we're trying to take them into the animal unit, show them what we do on a day-to-day, show them the importance of it, show them the care the animals actually get while in our hands. So we're trying to show those internal people everything that we do, but also we do lots of outreach programs, so going to universities, sixth forms, colleges, whatever that may be, and also presenting the importance of it, and again stressing the care the animals actually receive.
Yeah, I think that sounds great, because I think going into colleges and unis, these are the people that are going to be working in the industry in the next 5 and 10 years. So to spread that awareness, it kind of helps. I think just put people's minds at ease, right? That we're doing everything we can. We're heavily regulated. Some people from the outside world don't seem to see that. And I work at AZ too, and one of the drivers for the project, the business need for the projects I'm going to do is because investors that are completely non-science based, in AZ, banks and whatnot, put pressure on the company because they want to see that you're doing things, but they don't understand what we're already doing, all of these things that we do behind the scenes. So cheers for that.
Thank you. I think as well, with all the developing technology and silica methods and everything else, so you get the questions of why can't we just use those, but actually there is still a need, obviously, for animal research. So it's giving that understanding that yes, we are being careful as the 3 or the 4 R's as we're talking about now, but actually there is still this need, and trying to get over some of that controversy is difficult, isn't it? So Michelle has got her hand up, so I think might be talking about something similar later, so I'm sure she's got some questions for you.
It's really interesting because I'm going to talk about TrueCap.
Ah.
If you can stay on, I don't know.
That'd be interesting.
And the reason is because I proposed AKT as a drug target to CR UK back in, I won't tell you when, maybe in the tour. Yeah, and it was our compound that was licensed to AstraZeneca, which eventually turned into TrueCap. So, but I'm going to talk about team science. But the thing that I wanted to ask you, I'm just curious, what do you think is the most sort of exciting bit of your job? When do you get excited?
To a certain extent, I get quite excited every day because I mean, I don't just work outside of the animal unit. I only work on TrueCap, but inside of the unit, there's just a team of technicians, and we work across a range of projects, a range of compounds. And I think, like, when you see a compound so early on in its development, and you get to see in your hands the tumor shrink every day that you see it, I think it's really really exciting.
It's like,
really rewarding.
Yay!
Yeah. I think that's probably my favorite bit, is when you actually see the tumor shrink over like a matter of days, and then I think that's pretty cool.
Yeah, me too. Thank you.
Great, thank you. I think we have got a little bit of time, if you don't mind, James. I think there was another question from, there's a few more questions if you don't mind.
Cool. Yeah.
So there's one, did the PK/PD help explain the need for the dosing schedule? So for example, Cmax, Cmin, PD effect.
Yeah, so with the PK, we understood that scheduling all of our PD endpoints to the Cmax of the compound at 4 hours after the dose, I think that was really important. So lots of PK profiling. And then, in terms of the PD, we sort of learned that TrueCaps are used across a various number of cancer types, but understanding that like the same biomarker might not be applicable in one cancer setting compared to another. So then, in breast, it might be S6 or PRAS40, whereas moving to prostate, it might be something else. So it's I think it was said earlier, it's not always straightforward what you think it's going to be. So you're often looking at articles and papers published externally in a similar setting, trying to find what actually might be going on, because you can often see efficacy without actually quite understanding why, or it might be different to what you thought.
Yes, perfect. Thank you, James. And then there's another question, if you don't mind.
Go for it.
I haven't read this one through yet. Do you see often that in vivo rodent studies do not translate to human studies? So how do you generally prepare for translating rodent doses to humans? And do you understand why this translation sometimes fails?
In terms of translating our doses to human doses, it's not something that I've been part of specifically. We've got a big translational team that work out the bits from our preclinical to our clinical team, and they sort of sit in the middle of us. So understanding that, I can't comment on. There was other parts of that question, sorry, I've forgotten.
I think you did cover it. So translation to human studies, and then why does it sometimes fail? So I don't know.
Not sure I'm allowed to comment on that bit. No, I think I probably won't comment, just in case I say something I'm not supposed to.
That's fine, thank you, James. I think Michelle was saying she might be able to answer some questions, probably with perhaps, I don't know if that's later, Michelle, or if you want to provide some insight now.
I mean, I can say something about that now if you want.
That'd be fantastic, thank you.
I mean, there's a lot of experience about, that's why we have phase 1 clinical trials, because we have to do dose escalation. So we usually start at a very low, we know we predict what the dose is, and then we might start at even down as low as 2% of the predicted dose in a phase 1 clinical trial, because no matter how you predict, it is a prediction, and we use lots of mathematical models. I mean, I don't, the clinicians do. But, you know, so we always start, and that's why we have phase 1s. We can't start at what we think the calculated dose is, because it may not be the right dose. And so, you know, we have to consider risk for the patients, or the clinicians do. So often we'll start at 5 or 10% of the predicted dose, and then we'll do cohorts of 3 patients in a phase 1 clinical trial and monitor them. That's the short answer.
Brilliant. Thank you, Michelle. I don't know if any of the level 7, I might be putting you on the spot. Obviously, their first module is translational research. So I don't know if anyone wants to share some of their opinions or thoughts from the module, but I might be putting you on the spot there. Or if you want to share some of your thoughts in the chat for everyone to see.
There's
That'd
a
be
question
great.
from Samuel, actually, Kirsty.
From Samuel, perfect, thank you. So Samuel says, do you personally face any emotional challenges when working with animals in the preclinical setting?
Short answer, yes. You're working with sort of sometimes as low as sort of like 5-10 mice, sometimes up to 120 mice at any time. But yeah, when you work with the mice for sort of 8, 10, 12 weeks at a time, you sort of form a bit of a bond with them. So then, yeah, at the end of the day, like they're there for scientific purposes, and you want to make sure that they don't go to waste. So there's no point having the mice around and not collecting anything from them at the end of their life. But alternatively, or on the other side of it, you do form a bit of a bond with them. You've got to remember, yeah, you've got to make sure that you get use out of them rather than just having them around.
Yes, it's a good question. It's difficult. I've worked with rats mostly for many years, but I think it's difficult, and you do have to kind of try to draw a line. But at the same time, I think it is good that we still do feel that side of things. To, as you said, to then be careful, not start thinking them as big. We start thinking them of numbers in cages. I think it is good as well to help with those 4 R's. Also to try as best as you can to balance those questions and those things as well. So
I think I can weigh in on that as well. Like I request a lot of PK studies and PD studies, but I don't actually carry out the in vivo work, and so I can be detached from it. And then sometimes I just have to remind myself that actually, at the end of the day, there are these animals that are that are there. They're not just the numbers and the concentrations that I get out the other side. So I think it's really, really important to remember the ethical side, and I do have to sometimes sit myself down and remind myself that even though I'm just requesting it and then I get my samples back, there's this whole section in the middle that is really, really important to remember the ethical side of it.
I think a big part of like the sort of the emotional side of it is none of what we do is just me. All of my endpoints, all of my start endpoints, they're all sort of teams of 2-3 of us, because it's too much to take on as one person. But I think, yeah, like we do a lot of culture of care of the scientists as well as the animals, because I think a happy scientist have happy mice and vice versa. And so I think, like the culture of care of the people, making sure that they're emotionally supported, generally gets you better science anyway.
But also remembering, I think, just to add the regulation, I mean, the home office, the training that every researcher has to undertake. And I think it's changed over the last 10 years. It's become more strict. So really, anyone is working with animals now goes through really, really solid training, knowing what to do, how to do it well as well in the animal care. And that comes to the 4R, which is respect of animal, which I think it's really important and very well regulated in UK.